You're a Leaving Cert Biology student sitting your exam in June 2026. You need to know the Unit 2 chapter, Cell Division. So in the Cell Division chapter, you should already know definitions for the cell, which is the smallest basic functional unit of life, and continuity of life. Cells arise from other pre-existing similar cells. We also need to talk about the cell cycle. So the cell cycle depicts the life cycle of a cell. Now we have two parts to our cell cycle, a dividing phase and a non-dividing phase. The cell spends 90% of its life cycle not dividing, and that non-dividing phase is called interphase. So what's happening in interphase is the cell is preparing to divide, okay? So 90% not dividing. Then 10% of the time the cell is spent dividing, and that phase is called mitosis. Mitosis is a form of nuclear division where one parent nucleus divides to form two identical daughter cells. So the parent cell will be diploid, the daughter cells will also be diploid. They are identical to the parent cell. Mitosis happens in multicellular organisms like us. We use mitosis for growth of cells, for repair of cells. If we think of unicellular organisms, they use mitosis for asexual reproduction. So that would be functions of mitosis. Other definitions that we need to know. So we know that genes from DNA and RNA chapter, genes are sections of DNA and they're found on chromosomes. However, we don't always have visible double-stranded chromosomes. We only have visible double-stranded chromosomes during mitosis, when the cell is dividing. When the cell is not dividing, these condense into thread-like structures called chromatin. Still made up of DNA, still made up of proteins, still where we find our genes, just a different version when the cell is not dividing. They don't form visible double-stranded structures. Now what's happening in an interphase is the cell, I said, is preparing to divide. Mitochondria are replicating. If it's a plant cell, chloroplasts are replicating. We also have DNA and RNA, the nucleic acids, replicating. Proteins, carbs, lipids, all replicating, and protein synthesis is happening. So what's happening is the chromosomes are going to replicate. Now, in a cell, we have, this is one chromosome, but in interphase, that chromosome has duplicated or replicated. And these are held together at the region known as the centromere. But this is one chromosome. It is made up of two sister chromatids, and that's going to be very important when we look at our phases of mitosis. So in the non-dividing interphase, we have chromatin. In mitosis then, we have prophase, metaphase, anaphase and telophase in that order. Now, anaphase is 10 minutes, every other stage is 30 minutes. So what's happening is in prophase, the nuclear membrane breaks down, the chromatin starts to form visible double-stranded chromosomes made up of sister chromatids, or you can call it a replicated chromosome either, that's fine. So what happens is these start to become visible and the nuclear membrane, as I said, starts to break down. Spindle fibers are going to start to form, and they form from regions at both ends of the cell or poles of the cell called centrioles. The centrioles will form the spindle fibers. So we have prophase, 30 minutes. The next stage is M for metaphase, M for middle, because the chromosomes line up along the middle, also known as the equator of the cell. So what's happening is our replicated chromosomes line up along the equator of the cell. The spindle fibers pass through the centromeres, okay? So the centromere, remember, is holding the sister chromatids together in the pair. So what's going to happen is spindle fiber passes through the centromere and the chromosomes line up along the middle. Then we have anaphase. Anaphase, what's going to happen? Think apart. The spindle fibers contract, pulling apart our replicated chromosomes. Now what happens is those sister chromatids turn into daughter chromosomes when the spindle fibers contract. So the spindle fibers pull the daughter chromosomes to opposite poles of the cell, and that's anaphase. And then we have telophase. What's going to happen is the nuclear membrane reforms. The chromosome condenses back into chromatin. So we have chromatin here and chromatin here. We form two nuclear membranes again. However, the cell hasn't physically split in two. Because what happens next is cytokinesis. Cytokinesis, the splitting of a cell. And that's going to be different depending on whether you have an animal cell or a plant cell. So in an animal cell, a shallow groove called a cleavage furrow is going to form, and that's going to deepen until the cell divides in two. Whereas in a plant cell, a line of vesicles made of cellulose will form the cell plate down the middle of the cell and open a region called the middle lamella, and the middle lamella then will divide the cell in two. So that is your mitosis and your different stages in mitosis. Now we need to do definitions, haploid and diploid. Haploid refers to one set of chromosomes, n equals 23. Haploid cells would be the egg cell and the sperm cell. And as I said, n equals 23. If you were asked to draw a haploid cell, n equals 3, you would draw 1, 2, 3 chromosomes, n equals 3. If you were asked to do n equals 6, you draw 1, 2, 3, 4, 5, 6, n equals 6 chromosomes. Then we have a diploid cell. Every cell in the human body except the gametes is going to be diploid, 2 sets of chromosomes. These are your somatic or body cells. Okay, so these have 2 sets of chromosomes, 2n equals 46. Because we have 46 chromosomes or 23 pairs, okay, and they occur in homologous pairs. So remember, you need to have 1, 2. If you were asked to draw diploid number 2n equals 4, you would have 1, 2, 3, 4, is they are pairs. Okay, so 2n equals 4. Okay, so you have 1, 2, 3, 4. If you were asked to do 6, you would have 1, 2, 3, 4, 5, 6. Okay, with your pair. So that's your diploid. Then you need to know meiosis. Meiosis is another form of nuclear division where one parent cell divides to form 4 non-identical daughter cells. This is used by multicellular organisms for reproduction. It introduces variation. The parent cell could be diploid, the offspring could be haploid, so it halves the chromosome number. And as I said, it introduces variation. So that would be a function of meiosis. So that is the main part of the cell division chapter that you need to know. You need to know your interphase, your mitosis, your meiosis definitions, haploid, diploid, your chromosomes, and remember, your chromosome is replicated during interphase of the cell, and those sister chromatids are held together by the centromere, but that represents one chromosome. You also need to know about mutations, the likes of tumours, benign and malignant, but I'm going to stop there for this in this video, and hopefully that helps for you sitting your exam in June 2026.